Hi, DrBiology here, and this video is about vaccinations, and it's part of a series of videos on the topic of infection and response for AQA, GCSE biology and combined science higher tier and foundation tier. So vaccinations involve introducing a small quantity of dead or inactive forms of a pathogen into the body to stimulate the white blood cells to produce antibodies. So here we have a pathogen, and you can see on the surface of the pathogen are the antigens. So these are specific antigens on the surface. Now what happens is that these scientists will take the pathogen and they will destroy the pathogen and have parts or debris of the cell that contain the antigen. And that makes the vaccine, and the vaccine will be given to you through a syringe, through a liquid in a syringe, usually into the top of your arm, and it is injected into your body. It then circulates around your bloodstream, and that will stimulate your white blood cells to produce the specific antibody. So you'll notice the antibody will fit the antigen. Okay, and that means you have some form of immunity. So when you do come across the actual pathogen, if you're reinfected with the actual pathogen, your white blood cells can produce the specific antibody rapidly. Those antibodies will remain in your body, in your bloodstream, and in your lymph vessels for a long period of time. So if the pathogen enters the body, then the antibodies can be mass produced very quickly and rapidly, preventing reinfection. So let's summarize the series of steps in vaccinations. So number one, a small amount of dead or inactive pathogen is injected into the body. 2, the pathogen has an antigen on its surface. 3, white blood cells detect the antigen as a foreign body. 4, white blood cells produce antibodies which lock onto the antigen, destroying it. And 5, memory white blood cells are produced, which then means that if the pathogen re-enters the body, the antibodies are mass produced very quickly, preventing reinfection. So it's really important in a population that if you're going to have vaccinations, that a large proportion of the population are actually vaccinated for that particular pathogen to reduce the incidence of that disease. Now, one thing you might have heard about is something called herd immunity. So the disease cannot be passed on because it cannot find a new host and therefore consequently dies out. Now, the reason you need herd immunity is that some people can't have a vaccination. They might be too young, or they may be quite ill and therefore quite weak already. So therefore it's important to protect those people in a population through ensuring that as many healthy people are vaccinated as possible. So if you look at the infographic, it shows you the case when no one has immunity. So basically the contagious disease, the pathogen, will spread very quickly through the population. If you have some people who are vaccinated, then the pathogen can still be spread relatively easily between groups of people, and so some people will still be susceptible. However, if you get a large proportion being vaccinated, then the spread of contagious disease is contained. You might be asked as well to evaluate some information about vaccines. So this is a Dr. B's top tip today. So in the exam, you will be given plenty of information to analyse and evaluate. But here's some very common pros and cons of vaccines to help you along. So if we look at pros, vaccines have helped control lots of communicable diseases that were once common. So things like polio, measles, whooping cough, rubella, mumps and tetanus. They've also eradicated really deadly viral diseases like smallpox, and also polio infection has fallen by 99%. Obviously, epidemics can be prevented if large percentages of percentages of the population are vaccinated, and this gives us herd immunity, and therefore it reduces the spread of diseases that could be fatal or make lots of people very ill. The cons include vaccines don't always work, so sometimes they may not work, and sometimes they don't always give us immunity, so they're not 100% effective in everyone. The other issue is that viruses can mutate quickly, so you have to change the vaccine regularly, and many people need vaccinating on a regular basis. Some people may have a bad reaction. Now, this bad reaction might be slight side effects for 24 to 48 hours, so swelling or flu-like symptoms or even fever. Some people can be seriously affected by a vaccine, but these are thankfully rare, and there's lots of different safety checks that go on. So if you look at a later video of mine, looking at the development of new drugs, that will show what scientists do to make sure any vaccine or drug is safe to use. Now, I will mention the MMR vaccine because MMR is quite an interesting vaccine because there was quite a large, what I would say, controversy in the early noughties, so between 2000 and 2005. So MMR does stand for mumps, measles and rubella. And it's what we call a triple jab. So basically you get one vaccination with the three vaccines for mumps, measles and rubella. So in an exam you might be given some information. So here is some information about a study performed by Dr. Andrew Wakefield. And then you will be asked some questions. So I'm going to ask you to pause it, and I'd like you to please read this information, then we'll look at a question together. Right, so this is the type of questions you might be asked. So it does say use information from the passage above to answer these questions. The first question, was Dr. Wakefield's report based on reliable scientific evidence? Explain the reasons for your answer. So you would use that information to look at whether it was reliable. Well, it's for 2 marks, but you can see in the passage only 12 autistic children were sampled, so it's a very small sample size. The conclusion was based on hearsay, so the parents were blaming the MMR vaccine for autism. And in this case, only 8 parents linked autism to MMR, and Dr. Wakefield did not use any control. Next question, might Dr. Wakefield's report have been biased? Give the reason for your answer. Well, if you read lower down in the passage, you can see that he was being funded by solicitors and therefore the parents to do the study. So they had vested interest in finding out why their children had autism, and they believed it was the MMR vaccine. I'm going to add here, though, that in 2010, Mr. Wakefield was found guilty of serious professional misconduct, and he was struck off the medical register, so he was no longer able to practice as a doctor in this country. And I need to tell you that large-scale studies across the globe have showed that there is no link between the MMR vaccine and autism. Right. So let's go through a question checkpoint. So if you're new to these videos, what I ask you to do is to pause, read it and answer it, and then we'll go through the answers. So it's really useful for your exam technique to have a go at these questions. So here we go. So pause in 3, 2, 1. So this question is for foundation tier, but you can do it if you're in higher tier as well. So it talks about diphtheria, and it gives you some data on diphtheria and tells you when vaccinations began in 1941. So the first question, what evidence in the graph suggests that vaccination protects people from diphtheria? Well, you can see if you look at the data, there's a rapid decrease. So the decrease in the number of deaths. This next question, it says complete the passage by choosing the correct words from the box. So it says, during vaccination, harmless and then there's a gap. Now, they have got the words in the box. Now, this is an actual exam question. You'd expect it to say viruses, and I would expect it to say viruses. But if you look, the only word in this case could be bacteria. So is bacteria are injected into the body. Well, if it's a vaccine, it's usually for a virus. But I'm just going with what the exam question has said. So this causes white blood cells to make antibodies which help protect the body against diphtheria. Right, question 2. So again, I'd like you to pause, read and answer in 3, 2, 1. So this is a very common question. They love to ask you about MMR, and it says write down the name of 2 of these diseases. Well, you can have any 2 from measles, mumps or rubella. And then they give you some information to look at the risk of taking the MMR vaccine and not taking the MMR vaccine on children. And it says a mother is considering if she should have her child vaccinated. Use information from the table to persuade the mother that she should have her child vaccinated. So if you look, there's less or low or no chance of getting the named conditions, so the harm like convulsions, meningitis and brain damage in particular, if you are vaccinated. You could say that there's a quantitative figure, so I would use the data. So there's 5 times less likely to get convulsions, for example. Okay, so try and use the data in your answer for that second mark. Okay, this could be foundation or higher tier. So again, I'd like you to read it. I'd like you to, pause, read, and then answer. So in 3, 2, 1. So again, this question, it does say measles. What is MMR vaccine? Well, they mentioned measles, so the only other answers is mumps and rubella. Then it gives you some information and some data, and it wants you to calculate the percentage of the children who caught measles who had not been vaccinated. Okay, well, 504 have not been vaccinated, and there were 630 cases. So it's 504 divided by 630 times 100 to make it a percent, which is 80%. 2, suggest one advantage as a whole of, suggest one advantage to the population as a whole of children having the second MMR vaccine. Well, basically, there's a less chance of epidemic, or there's a less chance of spread of disease. What does a vaccine contain? Well, we know it's dead or inactive forms of the pathogen. And how does it prevent infection? So white blood cells produce antibodies. Antibodies are produced rapidly on reinfection, and antibodies kill pathogens. I think most people would forget that third point there about antibodies killing pathogens. Question 4. Okay, so again, here's one about data, using data. So I'd like you to pause, read and answer in 3, 2, 1. Okay, so they give you a set of data, and it shows them when the measles vaccine was introduced and when the MMR vaccine was introduced. And it says compare how effective introducing the measles vaccine was with introducing MMR. And it does say use the data. So let's talk about the measles first. So, well, measles and MMR, they both lead to a reduction. The measles vaccine does cause a larger drop. Again, you could use some of the data in the graph if you wished. MMR, though, does wipe out the cases of measles in the population. It doesn't fall as much as the measles vaccine, but that's because there were less people with measles than in the first measles vaccine. Okay, so again, I'd like you to pause, read and answer. So again, this is relatively simple, this question. So it's a describe what happens in the body after immunization. So again, white blood cells, they produce antibodies. And if you become reinfected, antibodies can be produced rapidly or quicker or faster. Make sure you get that word in about rapidly producing antibodies, or they're producing antibodies faster. Okay, question 5. This is definitely GCSE biology higher tier because it's about malaria and it's actually linked to a question on malaria life cycles. So again, lots and lots of information. So I will ask you to pause, read and answer in 3, 2, 1. So the keyword here is evaluate. So remember, when you see the keyword evaluate, that means the pros and the cons, and you've got to use the information that they've given you, and they've given you a hell of a lot of information. Now, a little top tip from me is that you could maybe take 2 coloured pens in with you, and you could underline the pros and underline the cons if you wished whilst you're reading it. That's quite a good idea. So anyway, let's have a look at the answers. So any 3 from the following. So an answer needs at least 1 pro and 1 con for full marks. There are lots of different things you could write here. So let's look at a pro. So it reduces disease, which is a pro. The success in the study with mice indicates that it might be successful in humans. However, as you'll probably know from the section on development of drugs, it needs to be trialled in humans before use, and you need to check the side effects as well. Another positive is the removal of genes prevents the parasite reproducing in the liver, and the injected parasite does stimulate antibody production. However, if you're injecting a live parasite, there could be a possible danger, and there may be possible liver damage because, again, it hasn't been trialled with humans. Right, okay, I hope you found that useful. It's quite a quick video today, and please do subscribe and put your bell alert reminder for more videos coming up shortly. I'll see you soon.